Okay, so let's take our second look at the instructions for life, and this time we're going to look at how those instructions get passed on, and that's in cell division. So, cell division is how complex creatures need to grow and repair their bodies. They need to make new cells. You began life as a baby, I assume, and you needed to get bigger, and so you needed to make new cells to do this. And cells make new cells by dividing in two, and to do this they need to copy the genetic information inside and pass it into each of the new cells. So there are two processes that we use for cell division. The first is mitosis, which is normal cell division, if you like, replacing cells and healing and growing. And this copies everything in the cell exactly, and it divides the cell into two identical daughter cells. That's what we call cells that come out of a cell division. And then there's meiosis, or meiosis, and this is cell division that prepares the genetic information for passing on. It recombines it and makes new cells which have half the amount of genetic information. So mitosis creates diploid cells, whereas meiosis creates haploid cells. So let's just mention interphase. Most cells spend most of their lives not dividing, and this is the time when they do all the normal things that they will do in helping you to live your life, and this is called interphase. So remember that before division starts and after it's over, all the cells spend most of their time in interphase. So let's start by talking about mitosis. So first, in mitosis, a cell will copy its chromosomes. So you will end up with a total of 96 chromosomes inside the nucleus, as they all get copied once. And this is called replication. So then these chromosomes are now in identical homologous pairs called chromatids. And pairs of chromatids then move to the middle of the cell, and they are pulled apart and dragged to opposite sides of the cell. And so now you have a complete set of 46 chromosomes at each side of the cell. And then you have the division of that genetic data into daughter nuclei and then daughter cells, and this process of division is called cytokinesis, which we'll talk about in a moment. So let's take a visual look at mitosis. Here is a cell nucleus with some chromosomes. Remember, there are 46 of these, so we've just shown a few, and those chromosomes will replicate to form chromatids, and those are little X-shaped pairs of identical chromosomes. And these chromatids will migrate to the equator of the cell, and spindle fibers will form. And these spindle fibers are like small wires that attach to the chromatids, and they can pull those apart as the cell divides, and that way you have one complete set of genetic information in each daughter nucleus. And those daughter cells are exact copies of the parent cell. They have exactly the same genetic information. So as we can see, each part of the process has its own name, and the first phase is called prophase, as the replication takes place. Then we have metaphase, as the chromatids prepare for division, and then anaphase, as the chromatids are pulled apart and the nucleus divides, and then telophase, which completes the cell division and produces two completely identical daughter cells. So let's go over again those stages of mitosis, shall we? Beginning with prophase, when chromosomes double themselves into paired chromatids. Then we have metaphase, where chromosomes line up at the middle of the cell and the fibers form that will pull them apart. anaphase is when they are pulled apart, and then finally telophase is when the cell division completes and you end up with 2 diploid cells, each with 2 N chromosomes. And remember that N is the haploid number, and that is the number of chromosomes you get in a haploid cell. So we mentioned before the process of cytokinesis, and after the chromatids have been split, the cell has to form a new nucleus and split in 2. And this requires a lot of things to be moved around inside the cell. So cytokinesis is a word which means cell movement, and it is a word that we give to the process of the cell rearranging itself inside to prepare for the final division. So we need to move on to meiosis, and here it gets quite a lot more complicated. So we need to be careful as we go through this. And meiosis begins in the same way as mitosis. DNA will replicate, and that will make chromatids. A total of 4N chromatids in each cell, and that is in 2N chromosomes. And it's important to remember, during cell division, a chromosome is what we call a pair of chromatids that is connected together. But after those chromatids are separated, each one of those in a new cell nucleus will be a chromosome. It's kind of complicated. So the homologous chromatids, after replication, pair up and swap sections of DNA, and this is when the genetic information in your mum was changed to create the egg that was you, and the genetic information in your dad was changed to create the sperm that was you. So after the homologous pairs of chromosomes have swapped or recombined their genetic material, they are divided into different daughter nuclei. So at this point, all of the chromatids are different from each other. Each one has changed some genetic information with a different part of the other ones, so there are no identical chromatids in any of the pairs. Each nucleus after the first division has 46 chromatids in 23 pairs. So this is the end of meiosis 1. But if you think back, we said meiosis was to prepare sex cells. And if you think back a bit further, we said that sex cells contain a haploid number of chromosomes. And the haploid number is just N. So currently we have 2 N chromosomes in each cell nucleus. What happens next? Well, there is another division, which this time is very much like mitosis. The chromatids line up in the center and are pulled apart into different nuclei, and this is the process of meiosis 2. So because we've already separated half the chromatids, remember we now end up with 4 daughter nuclei. So each of our daughter nuclei has only N chromosomes, which is half the diploid number, or otherwise known as the haploid number. And all 4 daughter nuclei have different genetic information. Not one of the chromosomes is identical to any of the others. So here is our cell nucleus, this time preparing for meiosis. The chromosomes replicate to form chromatids, and then the homologous chromatids meet and swap parts of their code. And so now you'll see we have each chromatid being a different color and all of them being different from the original nucleus. And then you can see that that nucleus with 4 N chromatids divides into 2 nuclei with 2 N chromatids. So the next stage is that the chromatids line up in the center and have a mitotic division, and at that point we end up with 4 daughter cells. And if you look at the pairs of chromosomes we have shown in each cell, they are all different colors from each other. So the daughter cells that we get from meiosis are called gametes, which we mentioned before, and these are the special sex cells that go into eggs and sperm. Pop quiz: how many chromosomes and chromatids are in each of the stages of meiosis? So if we look at the first stage in the top left, you make a quick guess and we will reveal that this cell has 46 chromosomes and 92 chromatids. And then the next one we see has 23 chromosomes and 46 chromatids. And then what about the gamete? The gamete cell has 23 chromosomes and 0 chromatids. And realize that at this point that is because what we were calling chromatids we are now calling chromosomes after the cell division is complete. Let's briefly cover the stages of meiosis. We didn't talk about them before, but they're all similar to the ones you know. Prophase 1 is when chromosomes double, and they also pair up into tetrads, so 2 pairs of chromatids is 4 chromatids. Tetra is a prefix meaning 4. Genetic recombination starts here, which is when the genetic material is swapped between chromosomes. And it continues in metaphase 1, where the genetic recombination completes and you end up with chromatids that are all completely different from each other. In anaphase 1, the homologous tetrads are now pulled apart to opposite sides of the cell. And in telophase 1, you divide into 2 haploid cells with 2 N chromatids. So these are called haploid cells because they only have N chromosomes, but at this point all of those chromosomes are doubled into 2 chromatids. And then we start with prophase 2, and at this point the chromatids prepare for the second division. They prepare to be divided into chromosomes again. And metaphase 2 is when the chromosomes line up at the center of the cell ready for this final division. Anaphase 2 is when the chromatids are pulled apart to opposite sides of the cell. And then finally we have telophase 2, when you get another nuclear envelope forming and the chromosomes are finally divided into 2 haploid cells, each with N chromosomes, each one different from all of the other daughter cells. So if we just put mitosis and meiosis together, the purpose of mitosis is growth and repair. The purpose of meiosis is to produce sex cells. Mitosis takes place in all cells apart from gametes, and meiosis takes place only in the reproductive organs. Mitosis produces 2 daughter cells, whereas meiosis produces 4 gametes. Mitosis creates the same number of chromosomes as in the parent cell, whereas meiosis creates cells with half as many chromosomes as in the parent cell. When we come to variation, mitosis has none, usually, except when mutation occurs, but basically the genetic information in mitosis is exactly the same. This is not the same as in meiosis, when daughters mix genetic material from the parent gametes. Finally, the variation between daughter cells. Are the daughter cells the same? Well, in mitosis they are identical clones of each other, but in meiosis all 4 of the gametes we produced are genetically different. So that gets to the end of the material we will cover, but as before we have Hank Green providing us a more detailed account of meiosis and mitosis, and you can check that out at Crash Course Biology on YouTube. These are of course far wider in scope than what we are studying. They are also not designed for ESL learners, so be aware of that.